your brain may fire one last electrical storm minutes after your heart stops. For most of human history, death was simple. Someone stopped breathing, their heart stopped beating, and a physician placed a mirror under the nostrils to check for fog. That was it. Death was a single moment, a line crossed. But modern resuscitation science and high-resolution brain monitoring have revealed something far stranger. Death is not a switch. It is a process, a cascade of events that unfolds over seconds, minutes, and sometimes even hours, and we can now watch it happen in extraordinary detail. The reason we now know this is partly because of a medical accident. Over the past 2 decades, doctors have brought people back from states that once were considered irreversible. And those patients, along with animal studies and dying brain recordings, have given us a window into the physiology that unfolds at the border between life and death. So let's walk through what the instruments actually show. Not near-death experiences, not folklore, not what someone felt. The documented biology, measured in volts and millimoles and partial pressures of oxygen, recorded by machines that do not speculate. It usually begins with the heart. When cardiac arrest occurs, the heart's electrical system goes haywire. Instead of the organized electrical wave that tells each muscle fiber to contract in sequence, the ventricles quiver chaotically. This is called ventricular fibrillation. Blood pressure plummets within a single heartbeat. The mean arterial pressure, normally around 90 millimeters of mercury, drops towards zero in roughly 4 to 5 seconds. That pressure drop is the trigger for everything that follows, because the brain sits upstream of every other organ in its demand for oxygen. It weighs about 2% of your body mass, but consumes 20% of your oxygen supply. It has almost no reserve, so when blood flow stops, the brain is the first major organ to feel it. Within about 10 to 20 seconds of cardiac arrest, a person loses consciousness. This is the point where someone collapses. It corresponds to the shutdown of the cerebral cortex and the thalamus, the relay stations that keep you aware and responsive. Neurons that were firing at a healthy clip just moments ago begin to starve. But here is where the story gets more interesting. The brain does not shut down uniformly. It shuts down in a specific, organized pattern. In 2013, a team led by researcher Jimo Borjajan published a study in PNAS that recorded rats during experimental cardiac arrest. What they found was not a quiet fade. In the first 30 seconds after the heart stopped, brain activity did not dwindle. It surged. That surge was measured in gamma oscillations, the fastest brain waves we produce, associated in living people with conscious processing, memory retrieval, and focused attention. The gamma power in these dying rat brains briefly exceeded anything seen during normal waking life. And it was synchronized across both hemispheres, a pattern that normally requires an intact, functioning brain. Now you might be thinking these are rats. What about humans? In 2022, an international team reported the first continuous EEG recording of a human brain during the actual process of dying. It was accidental. An 87-year-old patient was being monitored for seizures when he suffered a sudden cardiac arrest. The EEG kept recording, and what it showed was remarkably similar to the rat data: a surge in gamma oscillations in the moments before and just after the heart stopped. The surge was concentrated in the temporoparieto-occipital junction. That is a region associated with visual processing, spatial awareness, and the integration of sensory information. It is also, notably, one of the regions implicated in out-of-body experiences and visual hallucinations when stimulated or damaged. The researchers were careful not to claim this proves anything about near-death experiences. But the correlation is hard to ignore, which leads to a deeper question. Why would a dying brain surge? It seems counterintuitive. You would expect oxygen deprivation to simply silence neurons. The answer lies in the balance between excitation and inhibition in the brain. The cortex is held in a careful tug-of-war between excitatory neurotransmitters, like glutamate, and inhibitory ones, like GABA. When oxygen drops, the inhibitory systems appear to fail first. With the brakes off, glutamate floods synapses across the cortex. Neurons fire in waves, sometimes synchronously, sometimes in chaotic bursts. This is not normal brain function. It is a system losing its regulatory architecture. and it produces some of the most intense electrical activity the brain is capable of generating right at the moment when you would expect silence. But that surge is short-lived. Within about 2 to 3 minutes without oxygen, neurons exhaust their ATP. ATP is the molecular currency that powers every cell, and the brain stores very little of it. Without continuous oxygen delivery, ATP levels in brain tissue drop by roughly 50% within 1 minute and approach zero within 4 to 6 minutes. This is where the cellular catastrophe begins. ATP does not just provide energy. It actively runs the pumps that maintain the gradients across every cell membrane in your body. The sodium-potassium pump, for instance, constantly pushes sodium out of the cell and potassium back in, maintaining the electrical tension that lets neurons fire. Without ATP, those pumps stop. When the pumps stop, ions follow their concentration gradients. Sodium rushes into neurons. Calcium follows. The interior of every neuron, normally kept at a tightly controlled low calcium concentration of about 100 nanomolar, suddenly floods with calcium to levels 1,000 times higher. This is not a gentle shift. It is a chemical breach, and it is devastating. Calcium inside a neuron is not just an ion, it is a signal. It activates enzymes, some of which begin digesting the cell's own structural proteins. Proteases called calpains start cutting cytoskeletal fibers. Lipases begin breaking down membranes. The neuron is, in a very literal sense, beginning to digest itself from the inside. This is the point in the process that defines the window for resuscitation. For decades, the medical consensus was that brain damage from oxygen deprivation becomes irreversible after about 4 to 6 minutes. That number comes from the rate of ATP depletion and the onset of this calcium-driven cellular destruction. But recent research suggests the timeline is not as rigid as we once thought. A key discovery came from work on what is called the spreading depolarization wave. When neurons run out of energy and ion gradients collapse, a wave of electrical silence spreads across the cortex at a rate of about 2 to 3 millimeters per minute. It is called cortical spreading depolarization, and it was first described in the context of migraine auras. But in the dying brain, it is global and irreversible unless circulation is restored. This wave was confirmed in human brains in 2018 by a team led by Jens Dreyer at Charité University in Berlin. They recorded spreading depolarization in patients with catastrophic brain injuries, and the pattern matched what had been seen in animal models for decades. The wave moves outward from where energy failure is most severe, silencing the cortex as it goes. But here is the twist. The spreading depolarization wave, while devastating, is not itself death. It is a state of extreme vulnerability. The neurons are still structurally intact at this stage. Their membranes have not ruptured. Their DNA has not fragmented. They are silent, but they are not yet dead. This is what researchers call the penumbra, a zone of cells that are damaged but potentially salvageable. The actual death of neurons, the point of no return, comes later. It comes not from the oxygen deprivation itself, but from what happens when oxygen is restored. This is called reperfusion injury, and it is one of the most counterintuitive findings in all of resuscitation science. When blood flow returns to oxygen-starved tissue, the sudden rush of oxygen does not simply heal. It triggers a secondary cascade of destruction. When oxygen floods back into cells that have lost their ion gradients and are swimming in free calcium, the mitochondria try to restart. But they are damaged. Instead of producing ATP cleanly, they leak electrons, which combine with oxygen to produce reactive oxygen species, superoxide, hydrogen peroxide, hydroxyl radicals. These molecules are chemically aggressive. They tear apart lipids, proteins, and DNA. Simultaneously, calcium that flooded into the cells during the ischemic period now activates an enzyme called caspase. Caspases are the executioners of programmed cell death, apoptosis. They begin dismantling the cell in an orderly, choreographed way, cutting DNA into uniform fragments, packaging the remains into apoptotic bodies that neighboring cells can clean up. This is not chaos. It is the cell choosing a structured death over a messy one. This is why the timeline of death is so hard to pin down. The initial oxygen deprivation sets the stage. But the actual killing blow often comes minutes to hours later, during reperfusion, when the body's own repair machinery turns against itself. The longer the deprivation, the more violent the reperfusion injury. And the boundary between salvageable and unsalvageable tissue is not a wall. It is a gradient that shifts with temperature, age, and dozens of other factors. One of the most striking discoveries in this field came from studies on body temperature. When tissue is cooled, the metabolic rate drops, and the timeline of cellular destruction slows dramatically. For every 10 degrees Celsius of cooling, metabolic rate roughly halves. This is why targeted temperature management, cooling patients after cardiac arrest, became standard practice. It does not reverse damage. It slows the clock, buying time for the reperfusion cascade to be managed more carefully. There is also growing evidence that the process is not uniform across the brain. Some regions are more resilient than others. The brainstem, which controls breathing and heart rate, can survive longer without oxygen than the cortex. The hippocampus, involved in memory formation, is among the most vulnerable. This is why survivors of cardiac arrest sometimes have scattered, specific cognitive deficits even when overall recovery seems good. And then there is the question of what happens at the very end of the process, when resuscitation is not attempted. After about 20 to 30 minutes without circulation, the brain has passed through spreading depolarization, through reperfusion injury or the absence of it, and into a state where cells begin to rupture outright. Their membranes dissolve. Intracellular contents spill into the extracellular space. This is necrosis, uncontrolled cell death, and it is irreversible at the molecular level. But even here, the body has one more surprise. In 2019, researchers at Yale published a study in Nature that took brains from pigs slaughtered 4 hours earlier. They perfused them with a special solution called BrainX, a hemoglobin-based perfusate designed to restore circulation without causing the violent reperfusion injury we just discussed. And they found something astonishing. Neurons in these brains, dead for 4 hours, resumed metabolic activity. Individual cells took up glucose. Synapses responded to stimuli. The Yale team was extremely clear about what this did and did not mean. There was no recovery of consciousness, no coordinated brain-wide activity, no global electrical signaling. What they showed is that the window of cellular viability is longer than anyone expected, and the line between life and death at the cellular level is blurrier than the line we draw at the level of the whole person. The cells did not know the organism was dead. They responded to the right environment as if nothing had happened. So what does all of this tell us? It tells us that death, as measured by the instruments of modern science, is not a single event but a layered process. The heart stops, consciousness fades in seconds, the brain surges one last time, ion gradients collapse, a wave of silence spreads. And then, depending on whether oxygen returns, cells either repair, self-destruct in an orderly way, or rupture in chaos. The entire process can take minutes or hours, and we can now watch every stage of it in molecular detail. The line between alive and not alive, when you zoom in far enough, is not a line at all. It is a landscape. Subscribe to explore more of what science can now see at the edges of human experience.